This is TWIS, This Week in Science, episode number 530, recorded on Wednesday, September 2nd, 2015. It's Replicate Gate. Hey everyone, I'm Dr. Kiki, and this is This Week in Science. Today we're going to fill your heads with seahorse pregnancies, a runner's high, and a bunch of hot air. But first...
Disclaimer, disclaimer, disclaimer. It's time for a dose of reality. For millennia, humans have searched for the answers to life's eternal questions, and I have one of them right here. Why are humans different from other forms of life on this planet? The answers have been varied over the centuries. Some philosophers pointed to our creation and appreciation of art. Others our conscience, and yet more said it was our use of language or our superior intellect. And time again, science has shined a light on the natural world and then back at humankind and shown how perhaps we are not so superior. Perhaps we're not so different. Art. Elephants can create artwork and show preferences for some pieces over others. Conscience. Many social animals protect each other and act selflessly. Rats will rescue friends in peril, while monkeys will share food when they receive no reward. Language. Birds build songs just as we use words, and one African gray had a larger lexicon than most of us do. The last bastion was our intellect. We certainly have greater brain power than the beasts that roam the wild. But as we discovered last week, certain monkeys can master fractions better than many of us ever will. Fractions, people! So what is it? What do we have that the others do not? What makes us special? Are we even special? One thing stands between us and the wild. One thing has brought us our modern conveniences, our technologies, our complex societies, our medicines, and even our view of ourselves. The same thing that has also been used to prove we are not so dissimilar. Science. Science has taught us how much the same we all are, and in turn has set us apart. We do science, we live science, and we talk science on This Week in Science. Coming up next.
I've got the kind of mind that can't get enough. I wanna learn everything, I wanna fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I wanna know what's happening, what's happening, what's happening this week in science. What's happening, what's happening, what's happening this week in science.
Good science to you, Kirsten and Blair.
Good science to you, Justin, Blair, everyone out there. Welcome to This Week in Science. We are back once again to talk all about science.
My favorite.
That's right. That's what we're going to talk about. Before we get going, audio okay?
Yeah.
Yeah?
Oh,
That's
for
good.
us. No, to the audience. Who are we asking?
I'm just wondering if the audio is okay, because to me the music sounded a little bit weird. So, anywho, just checking before we get going. Anyway, and I can edit this out. Everyone, welcome. Thank you for joining us. This is This Week in Science, and we've got all sorts of science. I have brought today news stories about research replication, fat and microbes, and cancer-fighting wasps. What do you have for us, Justin?
A real-life Jurassic National Park being proposed. Tracking crocodiles in Wyoming and Canada. Clean coal that might make you glow, and CO2 into natural gas?
What? All right, let's talk about it. We're going to talk about it. We're going to talk all about it. Blair, what'd you bring?
I brought new insight into echolocation. I brought some bees. I brought male pregnancy and something we all need, sleep.
Always need the sleep. Everything we read about, everything always that we read about, it's, you should sleep more.
I agree.
It would be better if we slept more. Why don't we sleep more? So right after this show, I'm going to go sleep.
To bed with all of us.
But not yet, because right now we're wide awake. I want everyone excited because we're talking about science, and science is great, and science is always working hard, and science is always doing great stuff, and science is, oh, you mean, oh, that open science project where they were trying to replicate 100 psychological studies that have been done over the past decade or so?
Went great.
No.
Oh.
No, they failed to replicate more than half of the studies.
Half.
More than half. Unable to replicate the findings. And this is with the team of researchers, and there have been volunteer labs that have been volunteering for this project to help replicate. They've been getting help from the original producers of the studies, the people who originally published them, going to them and saying, okay, what did you do in your methods? How did you do it? Let's get this as exact a replication as we possibly can. And over half, unverifiable.
That does not look good, guys.
I think it's still better, just to keep it in a form of reference for this. And this is, I'm actually surprised they got 50% of them right, or to be able to reproduce.
Less than 50%.
Oh, okay. That's getting closer to what my number would have been. My number would have been about 35%. Because you're dealing with people. And whenever you're dealing with human psychology, there is this vast below the surface that refuses to conform to anything like a physical science would, and this is what you would kind of expect. So you would need multiple to even think you know what you're talking about. But still, more than twice as good as a financial advisor. Okay, and being able to predict financial market.
Yes, prediction and science are two different things.
Yes,
the predictions
they're not
science.
That's what I'm saying. Psychology hasn't completely fallen into the category of speculation based on these numbers.
No, it has not.
But it does show that it has high hurdles.
Okay, so here's the deal. So there were 100 original studies that were replicated. 97 of those 100 original studies had statistically significant results. In the biological and social sciences, that means a p-value of less than 0.05. Okay, less than or equal to 0.05, which is basically in the 5th percentile. So you have a 95% chance that your results are great and a 5% chance that you've possibly got a false positive or even a false negative, that there's a type 1 or a type 2 error going on in your results. So this means that in replicating them, even little tiny changes could potentially take something out of that 5% margin of error, right? So the teams that did the replications were able to get 39% of them statistically significantly replicated. So only, instead of 97, 39% are now statistically significant. An exciting thing about this is sometimes with social science and even biological sciences, it's the number of subjects that you have. So maybe the result seemed like it had a high p-value, but once you started actually adding in larger numbers of subjects and replicating, basically, okay, you did a certain number of subjects in this one trial, this one published paper. Let's try it again and do it again with this new number of subjects. Suddenly, it's a different story with the numbers of individuals, and so the statistics change. And you can get where something was a false positive. Suddenly, you start seeing that it was actually not a real result anymore. Additionally. Oh, go ahead.
Well, I guess part of it too is from what and I haven't looked at what you're reporting on specifically to see what kinds of psychological experiments that they were retesting. But one of the things that I've come across as a complaint to psychology is that the methodology that they use, even if they have a form of a double blind, that form of a double blind that they use in one experiment is not the same kind that's being used in another experiment, and a lot of the experiments themselves are set up almost ad hoc, or not to say ad hoc, but if you, I guess, with a lack of conformity across the field, which causes a lot of these problems. Where you're using
but in this particular replication effort, they're doing the best they can to completely exactly replicate the original study. So it's not, they're not just kind of doing something in a similar manner, where it makes it hard
exactly.
to do a meta-analysis.
And
they're
I think
actually replicating it.
Yeah, but I think that's the what I'm sort of getting at too, is that if you're replicating the same study and it doesn't come through, it's because the methodology they used to show that this is a result that came just from the experiment is unreliable. And it's, I think, largely because there isn't a coherent form of doing these studies that's shared throughout the field. So if each professor or group doing a study is coming up with their own ways of verifying their experiment, then they may be getting a data set or a result that won't conform to somebody else doing it at another time, or even doing it the same way again. There's a weakness in psychology in finding. It's much easier.
Okay.
Yes, I get it. It's much easier.
I get what you're saying, but I don't think it's just psychology either. I mean, what this paper hints at is the weakness of one part of the practice of science, which is that not enough studies are actually replicated, and we continue doing science based on single studies and single results. And so a single novel result sometimes is taken as like, wow, that's amazing, and it shifts a whole group of people into doing experiments along that vein and making assumptions based on that one single result, where in reality science and the knowledge that science brings is the result of many repeated studies, and results need to be verified again and again, and bigger numbers need to be brought in very often to actually verify that the result is a true result and not a falsehood in some way or another. And so, yes, I will agree that there are issues in how studies are done in the social sciences. I think there are scientists working to fix that. I mean, I hope so, that the social sciences and psychology become better sciences, because they could be better if the practice were much more rigorous. But this is something that goes across science, and what I want our listeners to get across is, like, even we report on a study. We're always saying, oh, this is a cool result, la la la, but we always couch it in the sense of, but we're waiting for more studies to actually verify this. Remember when we reported on this ages ago? This is, kind of linked to that. And so we always try and bring it into the bigger picture of science.
Oh, we were supposed to be doing that?
Yeah.
Yeah, so that's exactly what I was thinking about, and the fact that we've also reported on studies where what was supposed to be blind or double-blind studies have been proven where the results were skewed by the people doing the studies because they were expecting certain results. So that the fact that humans are doing the studies means they need to be replicated, because we are still humans who can influence data. Unless it's something that is measured by extreme technical instruments. And even in those cases, sometimes they're being read by humans, or the data is being synthesized by humans. There's always this need for replication, and on top of that, I think about the sample sizes all the time. We bring them up in our studies constantly, sample sizes. And this is a good reminder that we need to go back. We might think that we've read a study that is groundbreaking or tells us something brand new, and we can think about it that way, but we have to think about it with that asterisk of it might. Let's see more of this.
So, and here, can I rephrase what you also just said in a slightly more simplistic Tone. If you were doing a study on a group of animals and allowed them to self-report the data, people would probably have a problem with you allowing lemurs to self-report the outcome of the study. But this is what we do with the great ape, because we assume the great ape is always purely rational and concise and truthful and has a context for what they're saying when they speak words. But I believe nothing could be. We have to think of psychology as studying an animal and only being able to take as much data from that as we can get without the animal self-reporting. And that's what's tough, because most of psychology is about the self-reporting data.
So it would be interesting
So
to
much
see
is.
a set of 100 studies that weren't psychological studies that have not yet been repeated.
Replicated, exactly.
And
How will
see what the replication rate is there, to see if it's vastly different, if that one's 90% and this is less than 50, or if that's pretty much what it is, and that's why we need to replicate studies. I think that'd be very interesting to see.
And I bet you those that were replicated were also replicated in the West, and you could take them to another country and they would not be replicated, because this is only
Western
psychology
perspective. Yeah.
as it refers to Western psychology, correct?
It's a very good point, definitely.
Yeah. So from the Ars Technica article, and also from the original article in Science, the researchers ask, after this intensive effort to reproduce a sample of published psychological findings, how many of the effects have we established are true? 0. So that's basically what we're talking about. It's that it was one study, they replicated it, and so there need to be further replication.
Wait, they have to replicate the study where other studies weren't replicated in order to see if the studies were replicatable? Oh, no!
The Mobius curve of the scientific method.
Science, you have your work cut out for you.
Oh my gosh.
Yeah. And then let's talk about the end of the world. You know, the climate change thing. Yeah. Chlamydia, our planet's got it. We gave it to this planet, the chlamydia, and ever since we've just been like, well, how far can we take this? How much medication do we really have to give the planet? And the answer is, well, maybe more than we want to or we're hoping to. A new study out of ETH Zurich and also the National Oceanographic and Atmospheric Association Geophysical Fluid Light Dynamics Laboratory, they have used a very complex computer climate model, and basically that means that it has more parameters involved. When they say it's more complex, that means there's just more stuff in it. And it also, they let the model run over a much longer time frame than the Earth system models of intermediate complexities, or EMICs, that were used for the IPCC report, which gave us the estimate that we would have of how much carbon we could emit to stay below 2 degrees Celsius after we hit a peak. So the idea is, okay, if we stop releasing carbon, or we release a less carbon, if we want to stay below 2 degrees Celsius by 100 years from now and kind of have it peak there, this is what we have to do. And so these EMIC models, Earth system models of intermediate complexity, which actually didn't run that long and were not as complicated as this new model, they gave one answer. And so this new model has done a little bit more tricky math and running, and they determined that the atmospheric concentration of carbon dioxide, even though we might stop emitting carbon dioxide, the temperature, because of high latitude oceans and cloud cover that are not taken into the other models, the temperature will continue to increase above 2 degrees Celsius after the 100-year limit. So even though we do everything right according to IPCC, which we're probably not going to, it's still going to keep going up because there are other environmental factors at play. Blair, you have such a pretty look on your face right now.
It's not good news.
No, it's not good news.
However, I do want to add, this bad news does not mean action is pointless. What it means is that
we should do stuff.
we should do stuff to reduce our carbon. We're not going to stop it tomorrow, but this is just proof that we need to reduce it. And this is also a good indication that, yeah, our temperature is going to go above 2 degrees Celsius. It's going to go farther than we thought. And so, yes, that's bad news, but that means we have to get serious about our prevention and coping mechanisms that we have for what's going to happen. We have to really pour some money into research, not only on green energy, but on mitigation. Fixing our coastlines, finding ways to combat excessive storms and extreme weather. This means we really have to get serious because it's coming.
And to put a little emphasis on the doom and gloom side of this again, the 2 degrees does not mean simply that your local weather will be 2 degrees warmer. We're talking about a global temperature, which means your local temperature
could be significantly
higher. 10 degrees higher, right? It could be 20 degrees lower than the winter lows. We're talking about this is when they're talking about global temperature going up 2 degrees. Basically, mostly talking about ocean temperature, but they're talking about the global temperature as a planet. What that means for you locally is part of a massive weather system throughout the planet, where that 2 degrees over an ocean where your clouds are forming, or the 2 degrees over a desert where the air passed before hitting the ocean before it came. It's going to be more than a 2 degree change locally, I guess.
Well, and I think what's really important too is to think about the extreme weather events. That a change that drastic in global temperature means air and water currents are going to flip out. And basically it's just going to change everywhere. Everywhere is going to be different. And so I was just reading a study recently that said that San Francisco is projected over the next, I think it said 20 to 50 years, to have the same climate as San Diego has now. Which doesn't sound terrible to us.
But what does that mean?
It has implications that could be problematic. Not to mention that then San Diego will have a climate that is very different. And everything in between.
A lot of things are going to change. People are going to move. There's going to be a lot of things that we need to take care of. And we can't stop it now. We just need to be ready for it and do what we can to
We can slow it down, though.
Slow it down.
We can slow it down.
Yep. And so, Justin, can you I'm just going to go to your stories now, because you have one kind of good story in here.
Which I'm going to do last.
Okay, so you're going to do the bad news bears.
Well, actually, I'm going to follow up with exactly to the point of what we're just talking about here. From southwestern Wyoming to southern Canada, crocodiles have been found lounging in the shade of palm trees. Some of the strongest evidence of how drastic global climate warming can affect regional weather patterns. So there you have it.
We're not finding crocodiles in Wyoming now, though.
Yes, we are. Yes, we are finding them now.
Fossil crocodiles?
Huh? Oh yeah, they're fossils. Yeah.
Okay.
Oh, did I not say that yet?
Yeah.
Of course. These are fossils from 50 million years ago during the Cretaceous and Eocene era, when areas of southwestern Wyoming and southern Canada were very different climates than they are today. Exactly how, though, the middle of the North American continent, far from the warming effects of the ocean, stayed so temperate even in winter months is something that has sort of puzzled scientists. Like, even they don't understand. Even the biggest brains that have been put to this don't quite understand why it is, how it is, that we have these swampy-type crocodiles and palm trees in an area that has polar vortexes, right? So they, in recent decades, researchers observed those same high latitude regions in North America and Asia warming much faster than the rest of the world. New work by researchers at Harvard, Johnson A. Paulson School of Engineering and Applied Sciences, SEAS, and the Harvard University Center for the Environment, HUCE, suggests that increased amounts of low clouds in the Arctic due to rising Arctic temperatures could amplify winter warming in high-latitude regions. This mechanism offers a possible explanation to both past and future continental warming in winter. So this is,
Right.
So today the frigid air masses form as cool but temperate air from high-latitude oceans. They move across a sunless Arctic in the winter months. They get really, really cold. The air masses radiate heat out into space, cool strongly as the ice and snow covered over the ice and snow covered surface, then move south, which is what we normally experience in Wyoming and in southern Canada, that bitter, bitter, bitter cold air, Arctic air blowing through. Cronin and Zimmerman, who are the authors of the study, asked the simple question, would these Arctic air masses warm more or less rapidly if the high-latitude oceans over which they begin their continental journey were much warmer? So in the initial present-day scenario, ocean surface temperature is near freezing. They just called it 0 Celsius, using a very simple model here. They raised the initial ocean temperature all the way up to 20 degrees Celsius, mimicking the temperature during the Eocene and Cretaceous period. So,
and possibly what could happen if
possibly
what could happen with increased water vapor and changes in the CO2 and temperature.
We're still away. We've got the right CO2 levels for this already, but we have to go through some temperature change first. So in their simulation, when this air column moved over the Arctic and cooled, a thick layer of low clouds and fog formed. The clouds acted as insulators, significantly slowing the cooling process of this air as it moved over. Right. So all of a sudden, it's got that warmer air creates a blanket underneath it of fog, which insulates it from the icy tundra. So as it goes across, it remains warmer. At the end of the simulation, the surface air temperature was 40 degrees higher, despite the initial increase of only 20 degrees. Isn't that interesting? High-latitude clouds have a strong heating effect at the surface. We've all seen how a cloudy night cools off slower than a clear night. The same process is in effect here. Increased low cloud cover with warming would slow the formation of Arctic air. If the Pacific Ocean were very warm, low clouds could help these air masses and make it all the way across North America without ever dropping below freezing at the surface. The insulating effect could suppress the formation of frigid Arctic air altogether, says Cronin, which may explain the crocodiles in Wyoming. So
maybe we'll see crocodiles in Wyoming within my lifetime again.
Oh gosh.
Yeah. There's so much to look forward to.
So here's an example of what we were sort of alluding to earlier. Here's a 2-degree change in one location. That would be a 4-degree change somewhere else. You know, it would sort of it's
possibly even more. Yeah.
Yeah, and then if you get too much into it, you know, you think about the thawing of the tundra and the methane that's trapped in the ice, and then you have another catalyst for acceleration.
So there's a lot of variables. We don't know exactly what's going to happen. All we know is that it's going to get warmer,
and
weather's going to change, and dynamics will be different. Animals will move, plants will move. Let's slow it down.
Well, I think it's important, though. I mean, yes, we keep coming back to that, but I think it's also important to have these models, to have some idea of how things are going to change. I mean, the Earth is a very complex system, and there are many, many factors involved. And as over the years of this show, we have seen in reporting on these climate models that, you know, every year there's a new parameter that is introduced to the models, and sometimes even more than just one, to be able to explain how things happen and predict what's going to happen in the future. But we want to get as close as we can to estimating how things are going to change. We want to know where dramatic weather events are going to take place. We want to know where people are going to be able to live. We want to know where people are going to find water.
But the best way to learn that
predict this
is to allow global climate change to continue then, because this is this is like, for instance, how we're getting this data. They went and looked at it because there was this sort of anomaly. There's this central northern latitude region that seems to be heating up quicker than areas that you would expect, and so they went and looked at why that happens, and now they've got a nice rationale for why that's taking place. So I think as the ice cube melts, we're going to find out where that water's going to run, where that climate's going to change, and we're going to just get better and better at that prediction as time goes by. But it's still that I think there's one aspect of this, which is this is how we are going to prepare, because we know the rest of the great apes aren't going to do anything about it, which is fine. But the more important thing is a discovery made in Leiden that is helping not only make natural gas from CO2, but also to store renewable energy. Research by Professor Mark Copper and PhD student Jing Shen show how this process can be implemented in a cost-effective and controllable way. The conversion of greenhouse gas CO2 into natural gas is achieved using a chemical process, which CO2 is bubbles through an acid solution. The solution contains a graphite electrode to which a small negative voltage is applied with a cobalt porphyrin catalyst attached to it. It's already known that this catalyst can convert CO2 into carbon monoxide and methane, but the reaction has always released an unwanted element of hydrogen. In their investigation, Copper and Shen show for the first time how this process works. They therefore know exactly what the best acidity degree is in order to minimize the amount of hydrogen and convert as much CO2 as possible into natural gas. Added benefit, catalyst is entirely made up of common materials. Cobalt porphyrin is part of vitamin B12, while the graphite for the electrode is very similar to pencil lead. Therefore, the catalyst only costs... Yeah, it is very inexpensive ingredients being used here. Comparable methods of converting CO2 into methane often use rare and expensive metals, even platinum, which so it has a cost effectiveness built into their design. He hopes that their recent discovery will bring this dream a little closer to realization, to convert CO2 and water, the byproducts of fuels, into new energy or building blocks for the chemical industry. If this can be achieved using solar energy, you've done something even kind of cooler on top of it. You've created a way, not only in using solar for this, but being able to reuse that energy later. You've essentially created a long-term solar battery, a renewable energy battery, a storage method that you can have this gas stored. It's basically converting sunlight, in some way of looking at this, into a storable natural gas that can be used at a later date.
Right, and as opposed to just releasing the carbon dioxide, that would be the byproduct of these of these processes. Instead of just releasing it or trying to figure out some odd, convoluted engineering way of storing the CO2 under the earth, you can actually, if you can turn it into into natural gas, then you actually you maintain and extend the lifespan of the fuel that you've started with.
Right.
Yeah. And also, even though hydrogen might not, if they can make it more efficient, make the process more efficient by minimizing the amount of hydrogen, even if they take some of the hydrogen, hydrogen can be used as a fuel as well.
I know. I kept thinking that when I was looking. It's like okay.
If they can capture the hydrogen,
I get that you're going for natural gas. I understand that. But if you it's sort of like somebody, I'm trying to make coal, but all I end up with is this uranium byproduct. I've got to get this energy solution down to. It's like, I know you have. That's good too. Either way you run this, it seems like it's a win. But I don't. Yeah, but I don't know. I assume, based on at least what his attempts are to also maximize the efficiency of a renewable energy, that the hydrogen is not in a significant enough quantity to be utilizable compared to the natural gas that they're getting on the output, and that's why they're trying to minimize it. I would assume that doesn't state that in this article.
Yeah, all of this stuff, the technology is advancing to a point where it's becoming more and more efficient, which is fantastic. And we need this kind of research to show that it can be done inexpensively and that we have these processes available to us. The thing that continues to just be a problem in this field is the cost of fossil fuels. That the cost per barrel of oil is better at this point than anything else, and so it's competing against natural gas that we're already getting out of the ground. So why spend the extra money to turn the CO2 stuff that's coming off of that into natural gas that would then be competing against other natural gas that we're trying to get from other places? And so the cost aspect of it is what needs to be addressed. Mm-hmm. We can minimize the costs, make it more efficient, make it better, but at some point we need to step up as a society and just say, okay, we need to pay for this.
Yeah,
we need to include future costs into all of this somehow.
A good friend of mine told me once that in order for people to make the environmentally friendly decision in anything, it has to be easier than the old version. So her example of that was in a bathroom where the hand dryer was right next to the sink and the paper towels were across the room. And if you looked in the trash can, it was empty. And if you went to another bathroom where the paper towels were equidistant from the hand dryer, the trash can was full of paper towels. And that was her kind of example of you have to make it easier to do the right thing, which is how do you do that other than you need to make it clear to, I guess, your government that it's important to you and they need to put in the infrastructure. That's a pretty big uphill battle.
So part of this, though, part of the usefulness of incorporating the renewable energy to this is that renewable energy has storage issues. You can only have so many batteries. You can only fill so many batteries. You can lower the output of a coal plant by burning less coal, right? But when you have solar panels out there, they're just putting out power. Yeah. And if it's not being used right now and it doesn't have storage, batteries, whatever, huge battery banks, how many can you maintain? It just doesn't get used. So if this is a way to convert
solar into
solar that's not being used, the overages, you could just use the overages, apply it to this process, and you would end up with something that is long-term storage of that initial energy input that you put into it. And so it's a waste elimination. You could almost sort of think of it as a compost pile for solar and wind, where you get another use out of what would have been waste.
Yep. All right. Are you gonna? I think we should move it on up into the next step of conversation. Clean coal, or should we head into Blair's Animal Corner?
Oh, no. Let's go straight. We can save that for the second half.
All right, second half of the show, everybody. Stay tuned for more clean coal. But this is This Week in Science. Hey, if you listen to us on iTunes, consider writing us a review. Review This Week in Science on iTunes, or even just go in and say that you like reviews that are recent and helpful to you in liking the show. Do that for us. That'd be pretty awesome. Right now, it's time for Blair's Animal Corner.
Notes on features, right at stall. 5-peg, millipede, no pet at all. You want to hear about animals? She's your girl. Except for giant pandas and squirrels.
Let's
what you got, Blair?
let's talk about male pregnancy.
Woohoo! I've been waiting for this breakthrough for so long. I'm tired. Men, are you tired of waiting around for a woman to give you another child? Now you can have as many children as you want. Oh, wait, what?
If you're a seahorse.
In
Oh.
seahorses.
All right, Justin.
So it's the only animal that we know about that exhibits male pregnancy. And our understanding of it before was pretty much just that the females laid her eggs in the male. She put them in the male's pouch, and he just held them there, safekeeping, while they grew. Turns out male seahorse pregnancy is a lot more complicated than that. And in fact, it's pretty darn similar to human pregnancy.
Wait,
Let
what?
me explain.
You said what's similar? What?
Similar.
Seahorses?
Similar. So let's, for a second, think about human pregnancy. The reason our gestation is as long as it is because this little embryo, this little blastopore, before it's even an embryo, it has these little gas exchange wrinkles, and it embeds in the uterine wall, and that's how mom provides all sorts of things, nutrients, gas exchange, waste exchange, everything with the baby. And then later that's done through the placenta and umbilical cord, etc. It's a pretty complicated process in there so that the babies can grow big and strong before they come out. In fish, like seahorses, they have an egg. The egg has a yolk sac in it, and that provides nutrients. And they usually do gas exchange just with the water through a permeable egg shell is the
Membrane,
wrong word.
yeah.
Membrane.
Yeah.
So what it turns out, there's a whole bunch of gas exchange, nutrient exchange, and waste exchange happening through the male's pouch.
Wow, through the pouch?
Yeah. So
So it's like the pouch is a membrane itself.
yeah. So this is from the University of Sydney, and seahorses play as much a part in nurturing embryos during pregnancy as female mammals. And it's really wild because this is the first time that we've ever seen something like this outside of mammals. They're doing nutrient exchange, immunological protection, put an extra syllable in there, and they do gas exchange and waste removal. So all of this is going on in an animal that didn't even grow the egg. It got put into his little pouch, and he's getting all of that done. They also did RNA sequencing on the male seahorses during pregnancy. They monitored genes switching on and off during pregnancy, and they found that it was pretty much also the same as female mammals during pregnancy.
So were they gaining genes and with the young? So was there some kind of gene transfer happening?
That's interesting. They didn't
just
really
the
specify that. It was more about what was getting
hormonal profile.
Yeah, and what was getting switched on and off.
Okay.
And it also indicates that they used similar genes to manage pregnancy and produce healthy offspring as we do. So they were analogous in the
right.
in the
So
genes.
your body needs to not reject the babies. Your body needs to shunt nutrients to the babies. Your body. So what are all the genes that are responsible for this?
Yeah. So this
It's
is
interesting to know that the genes are similar.
Yeah.
Is this what do you call it? Convergent?
Convergent.
Convergent? Or
This is a convergent thing. So
or is it ancient ancestor?
well, then you would have to evolve out of pregnancy in the case of all amphibians, reptiles, and birds. So that's not so likely. The convergence is what's likely. What's so weird is that usually in convergent situations you see a huge difference in the genetics involved, and that's a good way to kind of clue into the fact that it's convergent. What's really interesting about this is that there's similarities in the genetics involved. So really what this shows you is that most likely the possibility for pregnancy is kind of just in there in vertebrate DNA. And so it's something very easy to happen or not, which is why it would be so easily a convergent trait. And if you think about it, it kind of makes sense, because if you look through all of the different types of vertebrates, all these different classes of vertebrates have their own little spin on pregnancy. So some of them have their super permeable eggs, some of them have their leathery eggs, some of them have their hard-shelled eggs, some of them have, like, in the case of marsupials, they have no placenta, but they're still pregnant, but then they have to have a pouch because they still have to grow their baby. So it's very interesting that you can see this kind of stepwise process. What's so weird is that with seahorses, they've kind of turned the other way. So it's very interesting, and it makes you wonder if good mother evolution has tried this at other times in the fossil record, and it hasn't worked out.
Yeah, when has it worked and when hasn't it? Yeah.
Very interesting.
So interesting.
Yeah, so male seahorse pregnancy, not so dissimilar from us. Also, here's another one. Bats. Bats are awesome at echolocation.
We love bats.
Right.
And all those stories about people with bats in their hair and all that kind of that rarely happens.
It's true.
And it's your fault if you have weird tangled hair for bats to fly into.
We think we know a lot about how echolocation works. We've plugged up bats' ears with wax and then tested them and seen what they do. We've done some brain scanning. We've done all sorts of stuff with bats to try to figure out exactly what's going on in their brain with echolocation. Now, this study from Queen Mary University of London had the first ever visual images from echolocation.
Wow.
So they tried to map how echolocation works, try to turn it into images. And this is what's really interesting, is that their understanding was that if you're a bat, you can echolocate and find flies in the air, but you can't find a fly on a leaf. And so they use scent, sight, other senses to try to find stationary prey on a surface. But there are some bats, there's some bats that actually use echolocation to find stationary prey on a surface. How do they do this? They use an acoustic shadow. So what they do is they actually learn the layout of a home range by their echolocation. So they come up with this memory of what the echolocation sounds like in their home range, down to every single surface, including individual leaves and rocks. If there's an insect on a leaf or a rock, the echo is interrupted or changed. That makes an acoustic shadow. And that is what's so strong of a signal to the hunting bat. And they found something that is kind of new called acoustic camouflage, which is when prey, they avoid this special kind of echolocation by finding a surface that disrupts the acoustic shadow. So what they found is that really smooth surfaces, like leaves and rocks, great for catching bugs. Rough surfaces, like bark, it's really hard for them to sense where an insect is. And so bats will trigger with their echolocation these extra smooth surfaces, because that's where they can sense where this prey is hiding.
That's so neat.
That's clever. There's a little bit when they said they were remembering what every leaf was like. That was a little, but I can get that they've learned that leaves generally should sound this smooth, and the problem with bark is that there can be enough modulation within bark that there isn't a clear signal of what bark should come back at that, say, a moth would really disrupt. But I get that the smooth rock or leaf, even if it was behind the leaf, could come back as enough of a noise difference from the standard leaf that you'd at least go and investigate.
Well, it's interesting you should bring up the moth, because the research team expected that bats could detect motionless prey better if the insects stood out more, if they had big wings, for example, like a hawk moth. When they rest, if the wing is down or up, if it's up, that would be easier for them to find, because that's a pretty big region that is being disrupted when they're sticking it out that way.
Yeah,
if they're sticking it out flat, it's kind of like
crazy.
A leaf.
Yeah.
But if they're like this, if they're straight up, then that's definitely going to change the sound. They found detection improves according to the kind of surface, nothing having to do with the insect's posture.
That's just fascinating.
Mm-hmm. So echolocation, the plot thickens, guys.
It's very complex. It's so fascinating that they can get such a rich picture of the environment.
Yeah.
From bouncing sound.
Yeah, so bats don't...
hearing the changes, yeah.
They don't look for prey. They search surfaces with which they are familiar, and in particular, smooth ones. And then they hone in on patches where the echo is weird.
And so maybe this explains getting tangled in people's hair.
Sure.
Because that would be a strange...
Yeah, that's a strange surface.
Result. Strange surface.
I wonder if they get caught in people's hair more if it's rough or if it's super shiny and silky.
Yeah.
And I also wonder if maybe there was a moth in the people's hair, but they just didn't know it.
That could be it too.
They didn't notice the moth, but they noticed the bat that came after the moth.
Well, the only way that would work is if that bat was hanging around long enough to know what your hair sounded like before the moth showed up. So, last quick story, bees self-medicating. So we've talked a lot about colony collapse disorder in bumblebees, and part of the reason for the colony collapse disorder is various parasites, particularly an intestinal parasite is one of them that's really rough on the bee colonies. And a recent Dartmouth-led study showed that bees infected with one of these common intestinal parasites, they're drawn to flowers whose nectar and pollen have a particular medicinal effect. So it was nectar containing nicotine and other natural chemicals in plants that reduced the number of parasites in sickened bees. And they had done studies on lab-made chemicals, including nicotine, that helped these bees. But then they just found that they do this already in the wild. So the particular concentrations that they were looking at was of iridoid glycoside compounds in nectar and pollen in 4 populations of turtlehead, which is a bee-pollinated wetland plant in North America. And they watched how the bees were attracted to these different types of plants, and then they manipulated the concentration of these glycosides, and then they saw how the bees' effects changed when they foraged, and they really found that the infected bees greatly preferred visiting the flowers at the highest glycoside concentrations. And ones who were attacked by something else, a parasitoid fly, not the intestinal parasite, did not respond at all in that way to the nectar. And hand in hand with that, they found that the flowers that naturally had the highest concentrations of the glycosides donated more pollen to other flowers, showing that their chemistry actually benefited their own reproductive success.
Oh, interesting. So,
yeah.
So having those glycosides, that chemistry, somehow allows them to produce more pollen, or maybe it's some metabolic aspect, but they produce a bunch of pollen and are able to get it around more. Well, additionally, I mean, if bees are going to those flowers more, because
Are they
make the
making more
glycosides,
pollen?
and then then the bees with that pollen on them are making more visits to other flowers, then maybe they're helping to spread that flower pollen around.
Yeah,
right?
so it's possible they just make more pollen because they get more visits, so they need more
Yeah.
pollen. That's what I
Yeah.
was thinking. But it's kind of impossible to tell what the chain of events was here that led us to this. But it does show that bees self-medicate, which is pretty cool, and that there are some wild plants that can help treat some of these parasites, and that potentially if we introduce the right plants into these habitats where these bees are found, maybe they'll medicate themselves. Wouldn't that be great?
Yeah. And maybe they already were, but a lot of those plants have been terraformed out of the environment.
Yeah.
And if this does translate at all to humans, it may be another reason to avoid smokers, as they may be smokers because they have a heavy parasite load.
Oh, the nicotine. Yeah.
Because of the nicotine.
Right.
But no, this is a good one of those things that, hey, if we've discovered a beneficial plant to our bee caretakers, let's put it in the medians. Let's start growing this in the in between highway lanes or wherever cities still water things to keep up the appearance of a lush town. Let's grow it there.
Yeah, well, I mean, there's the question with monoculture. You know, we've taken agriculture and turned it into this very monoculture-heavy state where we get rid of weeds, anything that and a weed I put in quotations because really a weed is anything you just don't want in there, but at the same time, a lot of natural ecosystems are, they are heterogeneous. They have lots of different varieties of things, and so over the years we're kind of getting rid of that, and that itself could be having interesting effects on the entire ecology of
on a micro example.
Exactly.
The city of Davis. All of the open spaces, the undeveloped spaces within Davis, as I recall as a child when I first got here, were overgrown in mustard weed. Mustard plants were everywhere. They were 4 or 5 feet high. I don't know that they were, but there was something perhaps that they were supporting in the local environment that now that they're not there. Even the empty lots are kept dirt. I don't know how they round up or what it is, but there's no mustard fields around anymore. And so you kind of wonder, well, what was that? Was that part of the local environment? Was there something that was relying on that?
Yeah, and when we reintroduce other plants, are there a bunch of subspecies that we don't know about, and the one that we're reintroducing is the wrong one? A lot of.
And I worry less. I think I tend to worry less about adding the wrong plant in, and I know that's naive of me, but I worry much more that adding just more of the same plant or of having nothing at all is a deleterious effect enough that putting plants, mixing up a little bit of what's out there and available would, I believe, have a better benefit to the ecosystem than, like you said, the mono culture plants or the decision to leave things bare. I think there's a benefit in diversity, especially in plants. Nowhere perhaps more so than in plants.
But I think the interesting point here is thinking of things. We tend to think of things on just, do we need the plant? How does the plant affect us? We don't think about it in terms of the entire ecosystem it exists in and how it interacts with other plants, how insects interact with it, how other animals interact with it, the web of life, Yeah. It really is a web. And so when you remove things and there's only one type of
plant.
Well,
it can handle full sun and limited water, limited drainage. We'll put that plant there.
And so this is just a great example of that, and it's,
Yeah,
great
and it's also a
example
great
of the
example
ecosystem.
of us thinking we need to step in and save the bees, but maybe the bees will save themselves if we bring back their plants.
Yeah, by self-medicating, which when you first said that, I was picturing a bunch of beers with, drinking, a bunch of bees drinking beers going, yeah, if I had the prescript, I'd be taking my pain medication, but instead I'm just going to drink.
Yeah.
They're
drinking pollen full of nicotine.
That's right. All right, everybody, let's keep working on this show. I'm going to go drink. Later.
Yeah, let's go to the bridge. Let's refresh our.
We're gonna refresh our beverages of choice. It's not vodka here. I'm drinking. It's water, people. Water is life. It is the end of the first half of This Week in Science, or I guess we could call it the first 2/3 of This Week in Science. We will be back in just a few moments with more stories. We've got bugs and guts and all sorts, and coal. I think we'll be back in just a few minutes. Stay tuned. Hey friends, this is This Week in Science. You listen to us as a podcast or on YouTube? Well, why don't you share us with your friends, write a review, recommend us in some way? That would really help us out. Do that. YouTube, iTunes, all these places for you to subscribe, share, and give us nice reviews. Help us get out there a bit more. Be a friend to TWIS. Also, if you like being a friend, you can wear our logo emblazoned all over everything. We have merchandise you might like. It's in our Zazzle store. So if you go to twis.org, that's TWIS.org, twis.org, you can find the Zazzle store. There's a link right in the main header. Click on it, head over to Zazzle, start buying. The holidays are coming. Oh my gosh, it's already September. When did that happen? Oh yeah, yesterday. TWIS is supported by listeners like you, too. Your donations help us do everything that we do. It helps to pay for contractors we need to hire to get stuff done, fun things we try to do, trips to New York maybe. We want to do that again this January. That would be awesome. Live show someplace. Let's make these things happen. Bandwidth, the basic back-end stuff. Your donations help, and we appreciate any amount that you're able to give. $2, $25. Please give. We accept donations through PayPal, so you can go to twis.org and check out those PayPal buttons all the way around the website. Go to just about any page, and they are there. Click and donate. Super easy. Alternatively, you can choose to support us through Patreon. Patreon is Patreon. Patreon.com/thisweekinscience is where you can find our Patreon account, where you can support us on an ongoing episode-by-episode basis. Whatever your preference, check out the website, listen to the show, make comments, and make a donation, please. If you can't afford a donation, like I said, it's very simple things that you can do, like sharing the show, writing a review. Your help to get us more people listening to the show will help us in the end, and it's one of the best things that you can do. We thank you for your support. We really could not do any of this without you.
And we are back with more This Week in Science.
This Week in Science! Dr. Kiki says, pumpkin spice, what? Whiz Mike in the chat room, no, it might be a brew, but pumpkin spice, it's only September.
Yeah, it's a little early, people. Come on.
I'm not ready for this yet.
I am totally ready for fall. I wish. You're just getting my hopes up. And I tell you, I've been thinking it's going to be fall since spring, because I don't think I got a winter.
No winter.
No. So, Justin, tell me a little bit. I mean, as fall comes along, we get cloudy air. Why don't you tell me something about the skies and coal ash?
Oh, this is a great study out of Duke University. Revealed the presence of radioactive contaminants in coal ash from all 3 major U.S. coal-producing basins. Now, we've talked about on this show before how the process of burning coal puts radioactive isotopes into the atmosphere. Because it's there, it's in the coal, it burns, it goes up into the air, and the data I've been told of says it's like the radioactive material in Fukushima being aerosolized above the United States every year. This study, though, was looking at ash. This is the stuff that is left behind from the burning of coal. The study found that levels of radioactivity in the ash were up to 5 times higher than in normal soil, up to 10 times higher than that of the parent coal itself because of the way the combustion concentrates the radioactive elements. Finding raises concerns about the environmental and human health risks posed by coal ash, which is currently unregulated and stored in coal-fired power plants, holding ponds, and in landfills nationwide.
Doesn't sound great.
Until now, metal and contaminants such as selenium and arsenic have been the major known contaminants of concern in coal ash, says Avner Vengosh, professor of geochemistry and water quality at Duke's Nicholas School of the Environment. This study raises the possibility that we should also be looking for radioactive elements, such as radium isotopes and lead-210, and including them in our monitoring efforts. Radium isotopes and lead-210 occur naturally in coal as chemical byproducts of its uranium and thorium content. Vengosh's research team revealed that when the coal is burned, the radium isotopes become concentrated in the coal ash residues, and the lead-210 becomes chemically volatile and reattaches itself to tiny particles of fly ash. This causes additional enrichment of radioactivity in fly ash. We've got radioactive enrichment processes taking place in this ash, people. Hey.
This is trouble.
Mm-hmm.
More coal. Let's put it more in the air. Awesome.
So another reason to hate coal. Got it.
Yeah.
Yep. Pretty much. Yeah, and this is thought to be, this is sort of the concept, this is sort of the work towards that cleaner coal is compiling this ash and sort of storing it in some sort of a way. But if you're inadvertently or unknowingly creating radioactive condensed regions that you're putting in holding ponds that can then leak, or landfills that can leak into waterways, you may be causing a bigger problem. So, at the very least, this study requires us to do some monitoring, I would think, if not regulating.
Knowing that there was radioactivity in the fly ash to begin with, we should have been regulating this and monitoring it more.
Well, technically, this is, I think, one of the first studies really to show that. I mean, this is, we knew it was in the being aerosolized, but I'm not sure they knew that these concentrations were taking place.
Right.
Right. So, because this is currently, it's unregulated.
Yeah.
What is done with coal ash? There is the President Obama's administration has put in U.S. Environmental Protection Agency has the first ever regulations on coal ash disposal set to go into effect this October.
First ever?
First ever, because this is just ashes. It's like we had a fire in our fireplace. Now we have these ashes that are left. What do? Stick them in the ground. Put them in as a compost bag, soil treatment. Like, who knows where this stuff has gone? This might be.
Well, it's probably gone into the ground, and then water. It's probably gotten into the water supply. It's probably leaked into. It's probably a large part of explaining some of the health issues that happen downstream from these plants.
Possibly. But it is, I think, another. Also, well, actually, one of the other things they want to do is they want to look at the workers.
They need to start looking at.
They need to look closely at the workers for radioactivity, because there's people who, I mean, they're now scrubbing the stacks and trying to keep the ash cleaning and condensed location. So you have workers there who are coming in closer contact with a 5 to 10 times higher than anything that they're normally in contact with levels of radiation. Yeah, this is something that definitely needs to get looked at, and maybe perhaps be added to the upcoming regulations that are coming up soon.
That'd be good.
Yeah. Not related to regulations, but kind of related to how our body regulates stuff. A couple of interesting studies out in the last week in a journal called Cell Metabolism. One looking at a hormone called leptin that is produced by fat cells, adipose tissue. Produces it, and it inhibits hunger. So the idea that we have this feedback loop wherein you eat, sugar and other stuff is taken up by the bloodstream, and it gets your body uses some of it for energy, and then other parts of it get stored, and the fat cells start producing leptin to inhibit your hunger so that you don't eat for a while until your body starts to use up its fat stores again, and then you want to start eating because you need to get energy, right? So anyway, these researchers looking at leptin, they developed a knockout mouse where, so basically they took out the receptor for leptin, for this adipose produced leptin in mice, and the mice that had this receptor knocked out, so basically the signal for inhibiting hunger would not get turned on because there's nothing for the leptin to connect to in these mice. So these mice ran around a lot more than their healthy, normal leptin receptor producing counterparts. So the mice that had hunger inhibited, they were just kind of normal active mice. But the ones that had none of the leptin connection, they started running around. They're using all sorts of energy. And so the researchers think that what this suggests is that leptin might actually be part of the signal that gives people a runner's high, or maybe is thought to be involved in anorexia, where people who have anorexia have hyperactivity sometimes. That basically what's going on is they have low fat stores, so they're not producing much leptin. Because they're not producing a lot of leptin, their bodies are trying to get them to run around to find food. So the idea is that it's not just running around, but it's running to try and get you to come in contact with food. So
here we go. This is it.
Maladaptive, interesting metabolism regulation.
This is the new diet pill then, a leptin blocker.
Yeah.
Wait, so
people are working on that kind of stuff.
The implication here is that the runner's high
is a motivation thing. That because people who run a lot, their fat is they're regulating leptin in a different way than the average person, because they're using up a lot of energy. So they may not be producing a lot of leptin.
So here it is, Blair.
So then they would be motivated to have more physical activity. So maybe it links into
so
the
body is telling them food.
Yeah, their body's telling them you're about to die. You've been running.
You've been running too long.
25 minutes.
You're going to die. You should get some food.
You haven't gotten anything. You don't have enough fat stores. I don't think we're going to make it through a cold night. Here's everything I've got to keep you going until we catch something or find something to eat.
That still seems counterintuitive to me.
Yeah.
But I guess it's because humans run with no eventual gain.
Right.
But the act of running is based on catching something.
Yes.
Yes, so your monkey brain, your ape brain, your animal tundra brain still thinks it needs to go and hunt and forage and eat something now, otherwise it won't survive the night.
Yeah, and so it's interesting also that it could potentially explain, like I mentioned, the hyperactivity in anorexics and people who are not eating, and that the hyperactivity could be the body's trying, it's not a straightforward signal It never is a straightforward signal. It's not like all of a sudden the brain goes, you need to go eat, go get some food, and forces you to the refrigerator. It's just your body trying to get you active in any direction that might have your body come in contact with food.
Right. And there are people who have eating disorders in the form of overexercise.
Yes.
Which I guess is not an eating disorder, but it's a disorder.
It is a disorder, but maybe it could potentially be linked in with this runner's high and leptin thing as well. So the couple of issues with the study that I have is that they're number one, they're looking at the leptin receptor. So the knockout mice were still producing leptin. They just couldn't, there was no receptor for it to link to, right? For it to have its signal completed. And when people are runners, athletes, or anorexics, they're going to just have decreased leptin production. They'll have plenty of receptors, but no leptin. So it's not the same. It's kind of similar, but it's not exactly the same. And then also to go from mice running around in a cage to this is why people's runner's high and anorexics are hyperactive. I'm kind of like, it's kind of a leap.
It is a leap, yeah.
But I like the leap. It's fun, and I hope it leads to more studies.
Interestingly.
And it didn't rely on any self-reporting.
It did not rely on any self-reporting, no. And then the second study that's also out of the same journal, Cell Metabolism, last week, really interesting. The researchers fed mice either a lard-based diet or a fish oil diet. And the mice on the lard-based diet gained a lot more weight than the fish oil diet mice. The lard-based diet mice, so they became obese, and then they started displaying symptoms for metabolic syndrome, and actually had markers for increased inflammation. And then the researchers did a whole bunch of other little tricks to kind of get to the heart of what was going on. But one of the interesting things, experiments that they did, is they took the microbiota from the different lard-fed or fish oil-fed mice and then gave them to mice that had been treated antibiotics. So they had no microbiota whatsoever. And then they fed them all lard. So antibiotic-treated mice were dosed with either microbiome from lard-fed mice or fish oil-fed mice. And then all those originally antibiotic dosed mice were fed lard. And the ones that had the fish oil based diet microbiome given to them fared much better, gained less weight, and did not have as many markers for inflammation as the ones that were given the microbiome from the lard fed.
I get that it accelerates. I wish they could have done it with giving them each a normal diet after, but I get that this sort of accelerates the result. That's fascinating.
Yeah.
So somehow the lard is compromising the gut microbiota.
Exactly.
Formation. Or just doesn't. Or just doesn't have it.
No, it's a combination. What?
It's the omega-3s, right?
Oh, I don't know if it's the omega-3s.
Last week we talked about how omega-3s aren't anything.
Well, they're not good for cognition, but they could be good for this kind of stuff. I don't know.
I mean, that is what they boast is in fish oil when they're selling the supplements. Yeah. Omega-3s.
But maybe they just don't know how. I mean, this is nutrition, so of course they don't know the mechanism. They just know perhaps they have correlated result of a healthy microbiome and better cognitive. And that can, if one is on a lard diet and
they haven't done anything with cognition. There's nothing with cognition.
No, not in this, not in this. But that's the claim of the omega-3, right?
Oh, right.
Sometimes,
which was
but
by
there's
itself,
other claims. They've made a lot of claims about omega-3s, and one of them also is processing your food, and I've heard things about omega-3s about you gaining less weight if you're taking omega-3s. I've also heard things about your immune system related to omega-3s.
So here's the interesting thing is that it may be the lard diet on its own triggered inflammation. It just with the lard diet they saw changes in particular signaling mechanisms related to inflammation. But then when they looked at the microbiome, they're completely different between the lard-fed mice and the fish oil-fed mice. And so that's the interesting thing, is that the microbiomes themselves probably potentiate whatever effect it is. And so so when they took the microbiome from the lard-fed mice and gave it to the antibiotic-treated mice and then fed them lard, it just potentiated, and so the mice gained more weight, there was inflammation, etc. But when they had the fish oil-fed mice and gave fish oil-fed microbiome and then gave them the lard diet, they didn't gain as much weight. Right. Because their microbiome was able to handle it. But over time, if you have fish oil for a while and then you just shift your diet to lard, that microbiome that you got from the fish oil is not going to stay.
It's going to crash. Mm-hmm. So it sounds like the fish oil is perhaps what the microbiome wants you to feed it.
Right.
I mean, this is sort of like,
maybe it's better. The fish oil is better for potentiating a healthy microbiome.
And the lard-fed microbiome is like, hey, knock it off. Hey, inflame something. Set something on fire. I don't care what it is. We got to get their attention upstairs. They're not paying any attention. They keep giving us this lard diet. Get us a tuna.
Yeah. So I don't know. It's really interesting. Really interesting. Not just your diet, but it's the combination of your diet and microbiome. And we've been talking about this for a while, and so it's just another piece of information in that direction.
Mm-hmm.
Yeah. Bronze Age brass? Bronze Age? Bronze? What?
Bronze Age brass. No buts about it. Billy O'Faglu from the Australian National University has found evidence that an artifact thought to be an Iron Age spear butt is likely a mouthpiece for an Iron Age horn. Talk about getting it.
Oh, that's awesome.
Back.
Words.
Spear butts word.
Yeah. Yeah, totally.
When Mr. O'Faglu used a replica artifact as a mouthpiece, the ancient Irish horn had a richer, more velvety tone than the version that had no mouthpiece previous. Suddenly, the instrument came to life, he said. These horns were not just hunting horns or noisemakers. They were very carefully constructed and repaired. They were played for hours. Music clearly had a significant role in the culture. Complex Bronze Age and Iron Age horns have been found throughout Europe, especially in Scandinavia. However, the lack of mouthpieces in Ireland suggested the Irish music scene had drifted into the musical Dark Age. I don't know what that would... Mr. O'Faglu was convinced mouthpieces had existed in Ireland and was intrigued by the so-called conical spearbutt of Navan. Though he could not gain access to the original bronze artifact, Mr. O'Faglu used the exact measurements to produce a replica using 3D printing and tried it out on his own horn. The addition of a mouthpiece would have given greater comfort and control to ancient horn players and may have increased the range of their instruments. However, few mouthpieces have been found. The lack of artifacts, though, may be explained by evidence that the instruments were ritually dismantled and laid down as offerings when their owners died.
I like this story.
No, this is because they found a number of instruments buried in bogs. The ritual killing of an instrument and depositing it in a burial site shows the significance of the instrument to the individual in that culture, right? So this is your John the Fiddler or whatever the instrument is. They bury you and they smash your fiddle up and throw it into the gravesite with you in the bog. You're buried with your flute in your hand. A number of instruments
because they found it and they were like, "Oh, this is for a spear. Yeah, that's it. It's for killing stuff."
Yeah. You can pry my French horn from my cold, dead hands.
Yeah,
but I mean that's sort of
and then it was for making music.
I do kind of like this for a very the personal reason. I was at Mesa Verde once, which is the Anasazi Indians, who have the the kivas and the cliff dwellings in Colorado and northern New Mexico. And as you walk into one of these things, they have this plexiglass display case, and there's like this sign that says unidentified artifacts. And I'm looking in there, and I'm seeing a whistle. It's got the hole in the right place. It's a whistle. It's a two-tone whistle. And I've seen this somewhere else at some other Native American exhibit, I think in Sacramento. So I know what this is. But then I realized they also know what it is. This is just sort of education bait. Like, oh, we have no idea what this obvious whistle is. Maybe you could identify it. And so I'm like, oh, I fell for it. But there is an element of somebody who's not a musician, if they saw a mouthpiece for a trumpet, might not know what the heck that thing is.
Wouldn't have a clue.
Wouldn't have a clue. It's a weird object, and hey, looks like it would fit ornamentally at the base of a spear. Yeah, so you could kind of stick it in the ground a little bit or something. You know, they got it backwards, they're putting it on the thing, right? And they're putting the horn end of it down into the ground as a little spike. Yeah, sure, it makes perfect sense. But yeah, then the musician sees it and goes, oh no, I know what that is. That's so obvious. It couldn't be anything else but. And when you look at this thing, it does. It looks very much like a modern-day trumpet mouthpiece. It's a little bigger, but it looks very similar to what's used today. This is also, he also throws into the end, you know, Tutankhamun had trumpets buried with him in Egypt. Contemporary horns were also buried in Scandinavia, Scotland, mainland Europe, and all of them had integral mouthpieces too. So the concept of having a removable, usable mouthpiece at the end of a horn wasn't at all unknown or unutilized. And they may already have had the artifact in their possession, just misidentified.
Just misidentified. Oh, look, I think it's a spear. No, that's a horn. It's most definitely a horn. Print it and play it, people. That's what we're going to do from here on out.
That picture you found, Kiki, that picture you found is actually, that's a better picture than the one I'd seen. Because the one I'd seen made it look much smaller. Now that I've seen it in the actual size that it is, because it was just against other mouthpieces at the time. Now I've seen that one. Makes it even a little bit more obvious, I guess.
Yeah. My final stories have to do with biological things that have potential. So one interesting study out this last week has to do with blueberries.
Blueberries?
Blueberries. I love blueberries. Blueberries are yummy, delicious, and they might have antibacterial and anti-inflammatory features, effects. So, publishing in the Journal of Agricultural and Food Chemistry, researchers took bacteria extract and used the extracts from the wild lowbush blueberry, Vaccinium angustifolium, against a bacteria called F. nucleatum. And F. nucleatum is one of the, or Fusobacterium nucleatum, is one of the main species of bacteria associated with gum disease, periodontitis. And these blueberry extracts inhibited the growth of the bacteria, and they also inhibited, to a very large degree, almost 90% inhibition, the ability of the bacteria to form biofilms. So one of the issues at hand is that, okay, you can get rid of some of the bacteria some of the time when you brush your teeth or with various compounds that you might use, but some bacteria get in there in the gum line and they form these biofilms that are very strong, very hard to remove. And how can you potentially either stop them from forming or get them to go away? And so this particular blueberry extract did a really good job, and it also blocked a molecular pathway involved in inflammation, which is part of a problem with gum disease as well. So they don't know exactly how well it will work in use, like using it on teeth, how it would be put into practice yet, but it suggests that there are aspects of the blueberry extract that might be really promising for gum disease. Blueberries, for gum
Eating
disease.
fruit to help your teeth. That seems
Ooh.
counterintuitive.
Doesn't it? Well, the fruit itself comes with a lot of sugar.
Yep.
So they're, eating
So
just
still
the
brush your teeth.
still brush your teeth.
So,
But there may be things in blueberries that could be helping your gums.
so part of the problems with sugar too is they also feeds microbes.
Yes.
But if blueberries, again, we know nothing. If blueberries happen to also be feeding the right microbes, then you could see an overall benefit perhaps to the gums. This is that field of which we know nothing. This is that world of which we just know they're there, and at one point assumed all were bad, and then some other point assumed that our main focus had to be kill every germ in your mouth, remove every bacteria from your skin, destroy all bacteria within your gut to get them out because they're germs. They're doing bad things. They're causing illness. And as we progress, we may learn more and more and more and more that actually we want to have a mouthful of the right kind of bacteria. And when we do, we certainly don't want to brush and alcohol mouthwash them away. Maybe brush, but not alcohol mouthwash
please
them
brush
away.
your teeth, everyone.
if we know we've got the right balance, because maybe it's hard to get that good balance back again. So we have our entire education still ahead of us on these issues.
Yeah. The other study that I thought was really interesting is that there is a wasp called Polybia paulista, and this story was sent to us from a TWIS listener, Dale. So Dale, thank you for this story. It's really interesting. Published in Biophysical Journal this last week, reveals how a toxin in the venom of this wasp, it's called Polybia MP1, kills cancer cells without harming normal cells. And I think we've talked about this before, but the question was, how does it do that? Why does it kill tumor cells why does it kill cancer cells? Why doesn't it kill normal cells? What's going on with this venomous toxin? Well, it turns out that lipids, so you have the cell membrane that holds in a cell, it makes a cell. And it's a lipid bilayer. If you've had basic biology, you know that there are these hydrophilic heads and hydrophobic tails, and so the lipid bilayer lines itself up and makes this nice little barrier to entry to just about anything. But it turns out that some of the lipids in the cell membrane are strangely distributed in cancer cells. Like they're just kind of not, they're a little weird. And so these researchers from the University of Leeds and also from Sao Paulo State University in Brazil looked at these phospholipids. There are two different kinds, phosphatidylserine, PS, or phosphatidylethanolamine, PE. So you have PS and PE phospholipids in the inner membrane that face the inside of the cell. But in cancer, guess what? They're embedded in the outside, the outer part of the lipid bilayer, and they face the outside of the cell. And it's the PS and the PE that the wasp toxin attacks. And they characterized it, and they found out that the presence of the PS increases the binding of the toxin to the membrane by a factor of 7 to 8. And then the PE enhances the ability of the toxin to rip holes in the membrane and increase their size by a factor of 20 to 30. So tumor cells, basically, because they've got these weird these weird phosphatines in the outer membrane that normal cells don't, they basically are writing themselves a death warrant when it comes to this toxin.
Yeah. Perfect.
Okay, I have a question.
Basically, okay, you're going to bind better and you're going to rip holes in the membrane better.
Right.
Yes, question.
So why does this toxin in the venom target that when healthy cells don't have that?
I don't know, but it could. I mean, it's a venom.
Happy accident.
It's a toxin. So
happy accident, maybe.
I doubt that. I mean, the fact that it is this wasp's venom it comes from, which is a defense mechanism or an offense mechanism for this social wasp. There probably are certain aspects of how that particular toxin interacts with cells of either other wasps or prey or attackers.
Either as a defense or an
something.
offensive mechanism.
Right.
I cure you of cancer is not one of the things that will.
It's something
This is just
happening
a
in one of
strain.
these other species that interacts with the wasp. Most likely, this is what it sounds like to me, is something that is helpful in the interaction with cells in these other species that by happenstance works out with cancer cells in humans.
Yes, by happenstance. Completely.
And how did we find this out? We were just plucking wasps out of the air and trying their toxins until one worked? Or
So I think
We're trying everything we can to fight cancer, and we're throwing everything possible at it.
throw wasps at the cancer wall.
Yes.
That's right.
Yeah.
Everything. Something will stay.
Well, there has been an effort by researchers to investigate various venoms to see how and why they have their have the effects that they do, because we are finding that there are uses for these venoms in human medicine. So, for instance, the cone snail toxin is potentially going to be amazing for managing pain. There are, lots of things that we are learning by discovering the properties of these venoms and the toxins that they contain. And so MP1, I'm reading here now, it says it acts against microbial pathogens and disrupts the bacterial cell membrane. So there's probably been a step-by-step research process that kind of went from, what does this toxin venom do? What's an active ingredient? Oh, it affects bacteria. What about this?
Yeah. And maybe that was it. Interesting.
Yeah. So people have been looking at this MP1. It shows promise for inhibiting prostate and bladder cancer cells and also multi-drug resistant leukemic cells. And so the question now is, now that we know how it works, how do we do further work to show that it really has the potential to be safe as a treatment to all people and not just animals in a lab or cells in a dish? So,
yeah.
biology, I love it. I love it. And then I guess we have some really quick stories for the end of the hour. My final story was pregnancy miscarriages could be linked to phthalates. So a new study of over 100 women, about half of them exposed to or who had miscarriages, and the other half-ish that were healthy women in China, looking at urinary phthalate metabolites, and higher levels of 3 particular phthalates were correlated with miscarriage. Doesn't prove it, but kind of good.
And where are phthalates from?
So phthalates are used in plastics, paints, medical tubes, vinyl flooring, soaps, shampoos, other personal care items, food packaging.
It's a lot of stuff.
A lot of stuff. Exactly.
Like most things in your house.
Yeah, exactly.
That's
so,
a bummer.
yeah, it's a bummer, but and there have actually been 6 of the phthalate substances that have been banned for use in certain products for young children in the United States already. So it's not as though we don't know about the risks, but this is just another possible risk. It isn't causation, it is correlation, but it's interesting nonetheless.
Yeah.
Yeah.
There's a proposal.
Yeah, what?
There's a proposal currently to create a real-life Jurassic Park. In fact, a Jurassic National Park. A Jurassic National Monument in Utah. Are we ready for this? Well, yeah, because the monument they want to make is in an area that is already sort of under some Bureau of Land Management control, because it is the highest density of Jurassic era fossils found anywhere in the world. And calling it a national monument will give it an added layer of protection that it doesn't have quite yet.
Hooray!
It is, yeah, so all for it. They should do it. No brainer there. Also, we may be in the wrong galaxy.
What?
Uh-oh.
Yeah, we might have taken a left when we should have taken a right. This is in looking at habitability zones within galaxies. They did this big study thing where they found that spiral galaxies, which we are in now, is not the type of galaxy that is considered the most habitable, because we have more supernova events. We have more things that can destroy your neighborhood and large neighborhoods around it than an elliptical galaxy, which is considered the most stable and the most, therefore, likely habitable type of galaxy in the universe.
All right, so we have some buddies waiting for us over there.
Now we know where to look.
More likely in the ellipticals.
Yeah, there we go. Start searching those ellipticals for deep space intelligent life signal.
Run the elliptical. That's right. And Blair,
So,
sleep as the last story.
how fitting before we all go to sleep. The long and short of it is that the timing of sleep is just as, if not more important than how much sleep you get. We've done a lot of studies on shift work and how that affects your body. Well, if you get the exact same amount of sleep, if you're a mouse, if you get the exact same amount of sleep, but at a different time than normal, you have a reduced immunoresponse. So
Reduced, so you don't fight back against things.
correct. Yeah. So you're not as good at fighting off stuff. You're more likely to get sick if you sleep at an unusual time. This is also things like upsetting your sleep, keeping yourself from sleeping, having your blue light from your cell phone near your face while you're sleeping. These are all things that could upset your sleep. So it's not about how much sleep you get. The point of this is it's about the quality, and that the mice in this study did not sleep as well, even though they slept as long, because it was at an unusual time and their circadian rhythms was off. So all of the things that could affect how good your sleep is, that's what's important, including when you're going to sleep.
And again, this study was done on mice.
Yes.
In the future, mice will unearth the archive of humanity, future sentient mice, and find everything they need to know about what goes on with mouse biology. And then we'll begin their experiments on humans.
I don't know. We've got a great experiment happening right now in the Nevada Black Rock Desert. I mean, Burning Man is happening. We've got 60,000, 70,000 people who are going to the desert and disrupting their sleep cycles. Let's just track illness after return.
Well, but there's other variables, like perhaps some mind-altering substances going on.
Yeah, and there's no greater mind-altering substance out there than a porta potty. That is the most. That is something that disrupts the mental state of any human.
Also, dehydration.
More so than sleep.
Dehydration, exactly, because
and ingesting too much sand.
It's not sand. It's dust, and it's special dust.
Oh yeah,
special dust.
Mind-altering dust.
But there's also a lot of light out there too. Massive light pollution.
The man, for example.
And noise pollution, and also
fascinating.
Sleep. So go to sleep, everyone.
There's a lot of wealth pollution that goes on there.
Go to sleep. Keep your cycles.
To sleep.
Keep your sleep cycle and your circadian rhythm strong.
But before you sleep, we will shout at you a bit. Because I believe before we end this program, we have some shout-outs to do.
Yes, and I was just about to do that. At the very end of the show, it's time to say thank you to our Patreon sponsors. Thank you so much to Kevin Donald Wesley Ballard, Ed Dyer, John Ratnaswamy, Steve DeBell, Gerald Sorrells, Greg Guthman, Dave Neighbor, Jason Dozier, Matthew Litwin, Eric Knapp, Andreas Oxstad, Jason Roberts, Patrick Cohn, Shana Tara Ginsburg, Richard Onomus, Byron Lee, E.O., Bob Calder, Jared Lysette, Ulysses Adkins, Brian Condren, Jake Jones, Trainer 84, Kati, Edvardus Rumkus, Brian Hedrick, Cassie Lester, Sarah Chavez, Stephen Soroyak, Layla, Marshall Clark, Charlene Davidson Henry, Don Kamarechka, Larry Garcia, Randy Mazuka, Tony Steele, Dave Friedel, Daryl Lambert, Kevin Parachan, Robert Francis Aston, Steve Mashinsky, Britt Kennedy, Phil Clark, Rodney Lewis, Braxton Howard, Phil Nadeau, Rick Ramus, Salgood Sam, Matt Sutter, Emma Grenier, Miranda McKennett, Philip Shane, James Dobson, Steve Goodwins, Kurt Larson, Stefan Insam, Michael George, Craig Landon, Russell Jensen, Mountain Sloth, Jim Drapeau, Darren Lute, Tara Payne, Alex Wisson, Johnny Schmaloney, Jason Olds, James, Noah Wilds, Paul West, Ed Bennett, Alec Doty, Illuma Lama, Joe Wheeler, Dougal Campbell, TMRO, Miko Pekala, Craig Porter, Adam Mishkan, Aaron Luthen, Marjorie, Paul Stanton, Scott Lajewski, David Summerly, Tyler Harrison, Bed Rothig, Colombo Ahmed, and Gary Swinsberg.
Yay, I give you all a virtual hug.
Thank you for all your support on Patreon. And if you are interested in supporting us, you can find information at patreon.com/thisweekinscience. And also remember, you can help us out simply by telling your friends about TWIS, doing a review on iTunes, sharing and subscribing to our YouTube account. And on next week's show, we'll be back broadcasting live online, 8 p.m. Pacific time, not Eastern, it's 11 Eastern, on twist.org/live Wednesday, where you can watch and join our chat room. Don't worry if you can't make it. You can find past episodes at YouTube, or actually twist.org/YouTube, or just twist.org.
Thank you for enjoying the show. TWIS is also available as a podcast. Just Google This Week in Science in your iTunes directory, or if you have one of those mobile devices, you can look for TWIS for Droid in the Android market sphere, or simply This Week in Science and anything Apple Marketplacey.
For more information on anything you've heard here today, show notes will be available on our website. That's www.twist.org, where you can also make comments and start conversations with the hosts or other listeners.
Or you can contact us directly. Email Kirsten at thisweekinscience.com, Justin at twistminion@gmail.com, or Blair at blairbaz@twist.org. Just be sure to put TWIS, TWIS, somewhere in the subject line, or your email will be spam filtered into oblivion. You can also hit us up on the Twitter, where we are at TWIScience, at Dr. Kiki, at JacksonFly, and at Blair's Menagerie. We love your feedback. If there's a topic you would like us to cover or address, a suggestion for an interview, a haiku that comes to you in the night, please let us know.
We'll be back here next week, and we hope you'll join us again for some more great science news.
And if you've learned anything from today's show, remember,
It's all in your head.
This Week in Science. This Week in Science. This Week in Science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth. And I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. Science, 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 science. This Week in Science. This Week in Science. This Week in Science. Science, 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 science. I've got one disclaimer and it shouldn't be news. That what I say may not represent your views, but I've done the calculations and I've got a plan. If you listen to the science, you may just yet understand that we're not trying to threaten your philosophy. We're just trying to save the world from jeopardy. Jeopardy! And This Week in Science is coming your way. So everybody listen to everything we say. And if you use our method instead of rolling a die, we may rid the world of toxoplasma gondii. Aye, 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 aye. 'Cause it's This This week in science. This week in science. This week in science. 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 This week in science. This week in science. This week in science. 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 I've got a laundry list of items I want to address. From stopping global hunger to dredging Loch Ness. I'm trying to promote more rational thought, and I'll try to answer any question you got. But how can I ever see the changes I seek when I can only set up shop 1 hour a week? This week in science is coming your way. You better just listen to what we say. And if you've learned anything from the words that we've said, then please just remember it's all in your head. Cause it's this week in science. This week in science. This week in science. 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 This week in science. This week in science. This week in science. 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 This week in 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 science.
Ha 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 ha. Nicely headbanged. Nice dancing there, Blair. Hello? Are you muted? You're muted. You're
I said
muted.
thanks. I decided to go for it. Full
You did.
throttle.
You did full throttle. What could I have here? Look what someone sent me. Or you, or Justin, but I got it. And then my child.
Is it the handy corn? Oh,
I got that. I did get that too.
I think that's supposed to be for me.
I got it. I have it.
Darn.
I have it.
I think Dave Friedel sent that.
Yeah, I went to the TWIS mailbox.
Those are so fun.
Look at these little guys.
They're awesome. Are those your color blindness glasses there? Blair?
No, I don't have $400.
Yeah, you do.
No, I don't. I have negative dollars.
I am Albert Einstein. I have come up with the theory of general relativity.
Come with the theory of your face.
I am Newton, and I discovered the law of everything. Thermodynamics, gravity, everything. Oh, wait. No, that was Darwin. Sorry.
Yeah, I was going to say that looked more like Darwin.
Newton! This is Newton.
No.
This is the everything dude. Principia.
Darwin's pretty sweet.
Here we have.
It's all right.
Ugh, get out of here.
I'm Charles Darwin.
I
I'm
was friends with the
Darwin.
tortoise.
I went on a boat and discovered change over time in species, natural selection.
I also wrote many essays on barnacle penises.
I love barnacle penises. They're such delightful biological occurrences.
Indeed.
Marie Curie, I was dosed with a lot of radiation. Hence,
Wait,
all my
what's that?
radiation discovery.
Mary, what's that in your hand? Now put that glowing rock down. Put that glowing rock. That's pure plutonium. Plutonium. What are you doing?
Oh my goodness, so fun. So fun. Yes, Whiskey Renegade, you were close. It was Darwin rather than Galileo. Yes. What fun. I don't know if these were supposed to go to Blair, but I have them. I'll give I'll give them to Blair at some point.
I just want my handy corn.
I have your handy corn. It's pretty awesome. Oh, look, wouldn't it be amazing if Darwin and Einstein could get their heads together?
Huh?
These finger puppets have magnets in their heads.
Oh my god, that's weird. So I was interviewed for a podcast on
tell me
Monday.
about that. Yes, tell me how it went.
It went very well.
I looked at the questions and I was like, nah, you got it. Fine.
So it's supposed to come out on Tuesday.
Cool.
So I will be tweeting when it comes out.
Awesome.
What happened?
It is called the Copeland Coaching Podcast, and she interviews people with interesting careers.
There, I've stuck them all together. Oh, I lost one.
The scientists got their heads together.
That's right.
In a
Interesting.
good
Yes. I would say you have interesting career path. I would think.
So we spent a little bit of time talking about the zoo, a little bit of time talking about the podcast. Hopefully it's helpful to people out there. I don't know.
I guess that counts as an interesting career path. Sure.
Yeah, like working with over 200 species and doing a podcast on air once a week and moving to Israel for 6 months. It's kind of interesting.
But that's all right. That's all right. It's up there.
Mm-hmm.
I don't know if it's up there with working
Selling
the
cars?
carnival circuit,
Oh.
then going becoming a door-to-door vacuum cleaner salesman,
I don't know.
then becoming a car salesman and a science news podcaster. I don't know if it's quite in the same.
Hey, it's not mutually exclusive. You could go on the show.
Well, I'm just saying, I don't never get invited to any of these fun shit. You guys are always going to the white tie fairs and everything else, and then when I go to an event, it's like, all right, let's see how well bleach actually can clean out a stain and vinyl. Like, I don't know how I get invited to those ones.
You could have come to the podcast movement, but you have these things called children that tend to get in the way.
These things. Yeah, my children are so massively in the way of my day that, you're right. I don't have time for anything.
But would you have time to be in SF Sketchfest if we applied and were accepted?
Sure. When is it, though? That's the thing. Because I'm really
January,
booked
February.
January, February.
Oh, yeah, totally. January and February are I'm open. I got nothing going on in January and February.
I think January, we may be going to New York again.
Oh, that would be awesome. Although, can we
When did we go this year? We went in January. I thought it was in March this year.
No, it was January.
What is January?
I think. Oh, wait. Was it? Yeah, it was pretty early.
Yeah, it was January last year. I thought you said they moved it to March.
They were thinking about it, but they didn't.
Oh, okay.
So
Great, let's
here's
do
the thing. Except, well, we'll talk about it later. But I think it would behoove my entire month of January if we stayed an extra day.
In New York?
Yeah, because we'll talk about it later. But there was some zombification.
Yeah.
Of trying to keep moving and
That's
keep
true.
not
It was
sleep.
rough.
Keep move. Don't sleep. Keep fly. Don't sleep.
It was a rough endeavor.
Wake up. Carry heavy thing. Carry heavy object. Make sure heavy object gets in taxi. Go. Go. Keep going. There was something a little intense about the pressure of.
It was intense. It was intense. I would agree. Add a day for sure. I would like to have had more than 2 hours sleep before we perform for an audience. Yes, I agree.
Although stunning. I love this. I love this in New York to the point where I'm starting to talk in Trumpisms. We were amazing. We were beautiful. We were amazing. We were top shelf. With 2 hours. I think with maybe 4 hours of sleep, we could have been even better. We could make TWIS greater. We want to have the greatness that is TWIS evident for everybody to see. And if we go with some extra hours, we will have that evidentness there.
Phenomenal. It'd be phenomenal.
Phenomenal. Yeah, we'll be heavyweight. Not lightweight. I'm not saying we were ever lightweight. I'm saying others, they come lightweight with a full night's sleep. With a full night's sleep, I think we would be the best heavyweight that ever spoke words with science out of their mouth at an event with other people looking at them while they said those words.
I speak science out of my mouth. Is that what happens?
Whiskey Renegade. Whiskey Renegade. Wait a sec. Somebody named Whiskey Renegade wants to ask me why I'm drinking out of a mason jar.
Hold on.
Whiskey Renegade. Your name is Whiskey Renegade. How can you call out anybody for drinking out of a mason jar? I'm just saying. Chat room.
Why does everyone have
room.
sunglasses
Live
on?
up to your name.
I'm so confused. I don't even own sunglasses, you guys.
I don't know. Blair started it, and I was like, she looks so cool. I'm not going to let her out-cool me. I'm going to go get my shades.
I gotta put my shades on. We're so cool. Awesome. What was I going to say? I forgot what I was going to say. Nothing. I wasn't going to say anything. I was just going to go back onto Google and try and schedule more hangouts. Must schedule more hangouts.
I speak science out of my mouth? That might be my favorite. I thought As the Ice Melts was great, but I think
what?
that's my favorite. I tweeted As the Ice Melts.
Like as the world turns.
Yeah, exactly.
As the sands through the hourglass.
Sands through the hourglass.
As each drip of water drips into the Pacific.
Different. Oh, we're like cornering
are the science
the market
news
on this. Because one was As the World Turns, and one was Days of Our Lives. One was As the World Turns.
As the grains of sand
like
drop in an hourglass.
So are the days of our lives. That we have left until global warming makes us move in with Canadians.
As the drips off a glacier into the Pacific, so are the days of TWIS science.
Because I speak science out of my mouth.
I speak science out of my mouth?
Mouth?
It was the...
Where does the science come out of? My mouth?
The emphasis that made it so glorious.
Mouth syllable.
Speak science out of my mouth?
Speak science out of my mouth?
So,
I'm excited.
If we go to Chicago, I'm told we have a place to stay there.
Hey, that'd be awesome.
Yeah,
so
if there's a Chicago event,
so that's where the podcast movement is next year, is in Chicago. And I'm already concerned about that because Chicago is much more expensive than Fort Worth.
Yeah, but I have like a familial connected
safe house for us?
I think art professory safe house first.
Safe house for us? Great.
Yeah.
We should go there.
Yeah,
let's wait to see if we get nominated again so our tickets are free.
Yeah, we should get nominated again. We're gonna try. I'll try that. I'm still on Spreaker. We'll see, or whatever it is. Was it Spreaker?
I don't know.
Spreaker or something.
I don't know. What's on my badge?
What's on your badge? Spreaker.
Also, I need to find out the absolute best satellite internet outfit in the world.
Wait, say that again? What? You need what?
If my dreams of starting my own church can come to fruition. I went and looked at this property today.
Oh yeah, the church,
today.
Right? Or something?
Yeah, or yesterday, I think.
Yeah, you're going to build a church?
It's already built. I just have to show up with some
renovate it.
Wheelbarrow full of money, and they'll be like, yeah, you can have it.
You're going to have to renovate it.
A little bit. But it's also rural.
Super rural.
It's rural, which means it doesn't have, the fancy modern internet connectivity.
Yeah, that's always a problem.
It has been. But I don't know. I haven't had, downloads, yeah, satellite's awesome. Uploads, satellite's far away when you try to upload to it. So I'm wondering if anybody out there has experience with this. I need to find the best possible. Yeah.
You're just gonna have to lay some wire yourself.
Well, yeah, I just gotta lay some cable 15 miles to Comcast headquarters.
No problem.
No problem.
Just
15
call, I think it's 811 before you dig.
Why don't you bring me that there cable stuff? Or, maybe you
Dig?
can bring
Who's
that
digging?
other
I'm just going to
faster
wrap
cable
that
that
sucker
you've
around
been
a power
talking
line.
about.
Oh, good.
Okay, cable. I'm rural. Come on, cable.
Right?
Come on, cable. Okay. Have there been any developments in your radio play? Re-record? Are you going to re-record it?
Oh, the most recent thing?
Yeah, or do I still just need to record something for you?
No, your recording's fine. No, your recording, everything was fine.
That's mine.
Actually, that's the editing.
Did I do it for you?
That's times. Yeah, you did. I think.
It's
Wait,
the whole
maybe
day we
not.
did in Davis that disappeared.
Oh, the other one I wouldn't worry about. Yeah, the other one I wouldn't worry about because you didn't do it, but everything that everybody else did who I needed was like white noise is what I got. So it's.
You need to re-record the next one. Okay.
Gotta redo it. But I'm still working. I just did some editing.
You're still working on the other one.
Yeah, but I'm really bad at this. I need to hire like an audiophile editor to go through and make this work.
Just do it for me.
I'm not good at it.
Make it happen.
I like to write. I like to direct. I like to even participate in the performance aspect of it a little bit, but I can't edit. I really am slow at it. Or I'm just bad at it. I don't know. It's hard to tell.
You're probably good at it. It just takes a while.
Yeah, and then you also have to have make yourself do things. You have to have discipline.
Yeah, there's that.
Born without that gene.
The discipline gene. Uh-huh.
So I look at this place. I look at this church, and I noticed it's got audio cables, or an audio, like a microphone, 3-prong microphone audio jack in the wall, like back behind where the pulpit would be.
Mm-hmm.
So I'm like, what? Okay, so this is interesting, right? So this place, I could set up a mic, but where's the output? Like, I don't know where the output is. I still actually haven't found the output.
There's speakers embedded somewhere.
The only big speaker that I've seen is this giant thing that looks like a loudspeaker. It's outside the place. It's out front. I'm like,
there's got to be more, or at least
maybe there's
somewhere.
Yeah. Maybe the speakers were removed. Maybe they took the speakers when they abandoned.
Are there organ pipes somewhere?
No, it's not this. It's not that elaborate of a church. But I have an electric.
Is there an area where it looks like there should be organ pipes, or would have been organ pipes?
No, there is not. But
like
this
a balcony
would have been a mic.
thingy?
There's no balcony. This is not. It's not a goth church. This is not a downtown San Francisco goth church. I'm sorry. This is a rural farm town church. Very different construction. But I'm kind of wondering, like, if it's connected to that loudspeaker out front, then I might be able to like hook up the mixer and a mic and like broadcast the show to a neighborhood of angry people.
Yeah, why don't you just plug in a mic and see what happens?
That's what I'm gonna have to do at least once. Find out. Funny. Okay, is there another end to this? Or if it is speakers, if it's speakers within the church, I mean, the place seems like it's small enough, but maybe not. Maybe but there's enough room to build some production studio space.
Yeah.
The problem is the live thing that we do is going to be hindered by being outside of the high-speed space.
Right.
It is problematic.
So, there's problematics involved.
Did I get 31, 32, 33, 34? Yay!
But there's a 1/4 acre of farm space if anybody wants to come up and grow some.
That's cool.
Be happy, whatever it was.
Would you live out there?
Tobacco, huh?
Would you live out there? Is that what you want to do?
Well, yeah, that's the whole. idea. Because it's got plenty of dirt to like grow big gardens.
Awesome.
And it's a nice small town. Like, I went and looked at the place, and the neighbors came out. I was like, "Are you gonna be my neighbor? Like, that would be great."
They're like, I
It's
don't
like,
know.
oh, really nice. Seemed like everybody was there was really nice, friendly people. Tiny farm town. Tiny town.
What town is
400
it
people.
in?
It's called YOLO.
Oh, yellow.
The town of YOLO.
Mm-hmm.
The city of YOLO.
Little. YOLO.
And I figured, I was thinking like, should I do it? Should I move there? And then I was like, I kind of want to do it. And then I was like, hey, YOLO.
Uh-huh. YOLO. Look at you guys in your sunglasses. YOLO. Jesus.
So,
Yeah.
never mind.
So. What? Never mind.
Okay.
I did another radio. I read for a radio play today.
Ooh.
Did you really?
Yeah, a short kind of chapter installation. It's a horror radio play, radio drama.
Nice.
Yeah, cool. I'm excited. I can't wait to hear it come out. Wait and see. I will share.
Oh yeah. I'm sorry. When I went to look at the place, there was somebody who was a representative of the church who showed the place.
Oh,
Uh,
did he tell you the scary things that happened there?
no. So I go. So the previous owner, you know, let the place go. He knows something that I should know? Because, I mean, I assume he knows everything. But if he saw the future of this property and said, you know, I don't want my people here. Is there a reason? Huh? Oh, no, I'm sorry. Well, I didn't specify. This had been a Catholic church. Therefore, women aren't allowed to be religious.
Right.
Except for
Women are just baby machines.
Yeah, but immaculately so. I mean, they do it better than anybody else can. I mean, yeah.
Unless you're a seahorse.
I am not a baby machine.
I would love to be a seahorse. Do you know how many kids I would have if I was a seahorse? I would have like 20 kids.
Yeah,
if I was a seahorse.
You would.
I would have a kid every
Have you ever seen what it looks like when a seahorse gives birth?
How many kids do they have at once?
I'm just going to send a link real quick.
Do this. Hold on. We can show a video.
Because I would just keep having kids.
All right, here, I'm going to screen share.
That's what I was going to do. Yeah. Just screen
Oh,
share.
by the way, again, Kiki, you screen share during the show, but I think you have to lock it on yourself.
I do.
Not just by clicking on it.
I don't think that it changes for you and Blair. I don't think the view changes the same way.
Oh, I would assume
for the audience.
Okay, we got to figure that out. We got to do a test. Oh, can we test it right now?
Here, look at
chat
this.
room.
Right now, I have it locked on Blair.
Okay, so chat room. Am I showing up in the feed as I talk, or is it locked on Blair's video of an octopi that you can't see right now because I'm continuing to talk and monopolize the screen?
It's not octopi, it's seahorses.
Oh, seahorse. I can't tell. It's in the tiny window. Is it sticking on me when I talk, or is it locked on the seahorses and I'm talking in the background? Somebody please tell us what is going on.
Okay, are you ready? Are you ready? Right now? Go!
Here comes the babies.
So many babies. Look at all those babies. Oh my gosh. Oh my gosh. Oh my gosh. Oh my gosh.
Oh, this is way more efficient than any woman I've ever impregnated.
Oh my gosh.
From now on, I wonder whatever genes that is. I'm going to start an army of me.
That looks terrible.
As Bjorkian as that can possibly be.
Look at all those little babies.
and you get a baby, and you get a baby.
How do they know
It
when they
is
get them
locked
all?
on video.
Come
Oh.
on, I'm not
So,
doing it.
so I stand corrected, Kiki. My concern that our feed convo flipping images was flipping around, even though we thought it wasn't.
Is it flipping around for you right
No,
now?
no. For me it is, but for me it's whoever talks is on what I see on
Okay.
screen. But according to the chat room,
It's on
locked
Blair.
on Blair's video, Seahorses.
That's weird. That's
So
weird.
you're right.
1,800 babies.
And you get a baby, and you get a baby.
Oh,
And
and
you
right
get-
after he
okay,
gives birth,
another
the
people.
female
You get another
impregnates
baby.
him.
You get like 2 more babies. Can you take? Who can handle like 10 babies?
And the female
Who
goes,
can handle
"Hey,
like 500?
you just had all those babies? Here,
Like
have
it says
some more.
Happy Father's Day at the end.
Have some more babies. Hey,
Oh,
Daddy,
gosh.
make some more babies."
Happy Father's Day.
Happy Father's Day. I'm going to remember that and send it to Marshall next year. Happy Father's Day. Aren't you glad this isn't what it's like?
So I haven't made any progress on the calendar this week, unfortunately.
It's okay.
But I'm going to try this coming week to get some stuff done. But I think that once we price it out, we can start listing it for pre-orders at least, as soon as we know what our price point is.
Yeah.
Because I mean, I have all the art. I just have to now find a place to print it. So, depending on how we want to take orders, I know we were talking about that before.
Yeah. Well, it depends on how we want to mail them out, what we want to do.
I have no idea what you guys are talking about, but apparently this conversation.
Calendriers.
Still, I don't understand.
It's all right. It's okay.
Blair's Animal Corner, calendars.
Oh yeah, that's a good idea. You know what I would definitely suggest though?
What?
Whatever we decide to do on it, we get it out well before January. Because
I want it to be
This is
listed by the end of the month is our
yeah,
plan.
'cause there's a certain point when people are like, you know what? I've already got one calendar, and come August, I'll probably realize that I haven't flipped it off of February. Like, like
I'm
as
very
often as-
good about my calendars.
Actually, I'm terrible. I always forget, but my kids are like, I got yelled at today. I was like, oh, Papa, it's already the 2nd, and you're still the last month. And they flip. So my kids are totally on this, and they're totally watching this thing. I forget the thing exists. So, but yeah, I think people lock into their calendar, the paper calendar, if they still have one, which I recommend everybody at least have one. They lock into it well before the new year. It's not something you go into February thinking, oh, I never got a calendar.
It's true.
I should do it now. They're not even in stores
do
anymore.
that.
You can't even buy them.
I had to buy it online last time.
Yeah. So get your calendar, get it early, get it Blair.
Here, I'll screen share these again. So here is a sample. Here we go. Look at this. See that right there?
I love the greens in that turtle.
And then you look at this. There's a vertical one. And then here's real quick. Good.
Head from Connecticut. January is the best time to buy calendars. They're cheap. Yeah, but only if you buy last year's.
Oh, here's the cover. I realized you haven't seen this yet, Justin, because you weren't here last week, right?
Yeah.
I still watch the show.
No. Well, yeah, so here's the cover. And then what I was looking for was man, can't find it.
It's somewhere.
I'm trying to find cripes. But well, here. Oh, I know what I'll do. Stop print. So I have to
Good
find
night,
a way
Kiki.
to.
I don't know. I want to take a breather.
Where did you go?
I think we learned that we need to go to sleep.
Take a breather. I want to go have a drink. Go to sleep.
I think we
Right.
should just all go.
Play with my magnet heads. Play with my magnet heads.
Okay.
So they have magnets in
chat
their
room.
heads.
Are you still there? Is chat room still awake?
Yeah.
Okay.
Oh no,
Question.
I don't know what my password is.
So everyone knows there's a man in a towel wandering around my house. Oh dear.
Okay. Account. There we go, my portfolio. Why is this so difficult?
Because it takes.
I don't know. What are you trying to do again?
I'm trying to just show an overview of the whole thing.
Just forget it.
Yeah. So everybody buy the calendar. Kiki, go identify the man in a towel wandering around your pad there. And I am I'm gonna go do some cabaret.
Cabaret sounds awesome.
Yeah, I gotta change. There's apparently a frilly dress, kind of a big tutu-looking thing, and some nylons I gotta slip into. And I don't think I'm gonna make it in those heels, but I'm gonna try.
I'm confused.
Yeah, you should be. Good night, everybody.
Good night, everybody. Thank you for joining us once again. We're going to work on that calendar. We're going to work on all sorts of fun things for you.
Yeah. Yep.
Thank you. And we'll be back next week, as usual, Wednesday night. In the meantime, if you're still watching right now, check out the TWIS Minion Science Island Hangout tomorrow night. You can go to scienceisland.org and you should be able to find out info there. Science Island for the win. But yes, waiting for the Vogon spaceship. That's right. That's what we're doing here, everybody. Have a good one. Enjoy your Wednesday. The hump day's over. Enjoy the rest of the weekend. We'll talk to you later. Bye! Stop broadcast. Don't just leave call. That's what I